Hi everyone, welcome back to this YouTube channel. The topic for today's video is going to be blood typing and blood smears. We are specifically going to be looking about the laboratory experimentations involving these two techniques. The aim of this presentation is to cover these two techniques in an orderly manner, involving the materials required, procedure and lastly discussion. The first part of this video is going to deal with blood typing. We are going to first begin by a few basic questions such as what is blood typing and why is this procedure carried out. The purpose of this experiment is to identify the individual blood types. Based on the antigen-antibody reaction, we will be able to determine an individual's blood type. Through using the anti-A, anti-B and the anti-D serum, we will be able to ascertain the specific blood types of each individual. A sample hypothesis for this experimentation can be, based on the antigen-antibody specific reaction, we can determine the ABO and the RH blood types using specific anti-serum. This is due to the fact that if any A antigen comes in contact with an anti-A antibody, the blood will undergo a reaction which will clump the cells together, and this process is referred to as agglutination. If no reaction is caused in the blood, then the individual has the blood group O. The materials required in order for you to carry out this experiment are microscopic slide, anti-A, anti-B and anti-D reagents, ethanol, marker, lancet, hematocrit tube, toothpick, 3 small toothpicks, cotton, tissue and bandage. Set the clean microscopic slide on the white paper. Place 1 small drop of each reagent side by side on the slide and label the drops using a marker. Sterilize a finger to be pricked using detergent and ethanol. Prick it with a lancet using a firm, rapid motion, or a blood sampling device. You must lacerate some capillaries so that the blood flows out. Collect the blood in a hematocrit tube. Once it is in the tube, it won't clot due to the heparin coating. Hold a cotton on the wound to staunch any residual bleeding, can also apply a bandage. Shake out a drop of blood next to the drops of the reagents. Stir the blood into each reagent with the end of a clean toothpick. Use different toothpick while mixing the blood with the reagents. We have 3 reagents, so prepare 3 different toothpicks for each sample. Wait for 30 to 120 seconds to observe if agglutination will occur or not. Anti-D, which is the RH factor antibody, is particularly slow to react. From the results, when the blood mixed with anti-A antibodies and anti-AB antibodies, blood agglutination was formed, meaning that in the blood there was an antigen which belongs to blood type A. We could say that this blood was blood type A because although it reacted with anti-AB antibodies, it did not show any reaction in anti-B antibodies, which always reacts with B antigen. And if it were blood type B, the blood would not be agglutinated when mixing with anti-A antibodies. Thus, in the blood, there was only A antigen that could identify it was a blood type A. Also, it could not be blood type O because there would be no changes, no blood coagulation, if it were blood type O. From knowing this, it would be dangerous if blood type A were injected to a person with blood type B, or if injected blood type A or B to a person with blood type O. The next topic of discussion is going to be blood smears. This is also going to follow along the same format and will involve detailed analysis on the procedure as well as the results and discussion. The materials required for this procedure are microscopic slide, ethanol, marker, lancet, hematocrit tube, microscope, Giemsa stain, methanol, cotton, tissue and bandage. A single smear can be made per slide, smearing running the length of the slide. Start by taking 2 clean microscopic slides. By using a marker, label your initials on the microscopic slide. Collect blood in the hematocrit tube. Only 1 to 2 mm is needed. Blood should be placed on the edge of the clean slide and you should avoid touching the slide to the skin. The angle between the spreader slide and the sample slide will vary depending upon the size of the drop and the viscosity of the blood. The approximate angle recommended is 30 to 40 degrees. The spreader catches the drop and it spreads by capillary action along its side. After smearing, let the slide air dry. Then dip it into methanol for 20 seconds. It fixes the smear by dehydrating it and denaturing the proteins. Then air dry the methanol. It also helps make the cells more visible. Immerse the fixed smear in the Giemsa, and then leave it for 20 to 45 minutes. Rinse very gently under tap water. Examine the slide under high powers, culminating with oil immersion. Add a cover slip to observe with oil. The cells can then be located under the microscope. In this experiment, we did blood type testing with considering how the blood would change when it was mixed with each antibodies, anti-A, anti-B, anti-AB. and we indicated from the changing what blood type it was. The change happened because of antibody cross-linkages established when one antibody reacts with antigen on two different red cells. Antibodies bonded with its corresponding antigens to destroy them, as when B-type blood is injected into the one with blood type of A. The anti-B antibodies present in blood type A will interact with antigen B from blood type B, forming blood clot. That blood clot can bring an individual to death because when the substances get out from the cracking of agglutinated red blood cells, they turn toxic. Hence, it is very necessary to know our blood type and which blood type can match with ours. A person with blood type AB can accept all blood types. A person with blood type A can receive blood from. Thank you for watching this video, and please subscribe to this channel for more videos in the future.